Okay. So in the previous lecture, we talked about the microtubules, right? So based on those microtubules only, your cilia, your flagella, okay, which helps in the locomotion of many bacteria, the sperm, it is helping. So we are going to see like how with the help of the motor proteins and the microtubules, it is helping, it is propelling, okay, and helping the cell to move ahead. So we know that like how there is a sperm is there, and the sperm has got this flagella, and you know that it propels so fast that it moves, right? It swims very faster. So we are going to see like how this the microtubules and these motor proteins are helping them to move fast, and have got the videos also where you can see that the cilia of, for example, the what do you have, the organism paramecium. So paramecium is there, so they have got the cilia in order to move, in order to sense, right? So we're going to see like how with the help of these microtubules it is helping them for the movement. Okay, so basically if you see, actin filaments and microtubules are meant for your cell movement. Okay, that is their major work. Now, cilia and flagella are microtubule-based projections of the plasma membrane. They act as antenna. Cilia act as antenna and sense a variety of extracellular signals as well as responsible for the movement. It should be noted that many bacteria also have flagella, but these prokaryotic flagella are quite different from those of the eukaryotes. Now, bacteria flagella are protein filaments projecting from the cell surface rather than projections of the plasma membrane supported by the microtubules. Did you get this? Bacteria ka kya hai? They have got the cell wall, right? So they will be projecting from there rather than by the cell membrane. Whereas in the case of the eukaryotes, it is from the plasma membrane. Sorry. So there are two types of cilia: primary cilia and the motile cilia. So what is the difference between the primary cilia and the motile cilia? नाम से ही पता चल रहा है that they are not meant for the movement. They are meant for the getting the signals, chemical signals from the environment. Okay, so they're involved in sensing extracellular signals, including movement, odorants, and the light. Whereas motile cilia is meant for the movement only. For example, odorant receptors in the olfactory neurons and photoreceptors in the retinal cells are located on the primary cells. Now, when you talk about olfactory neurons, this is meant for what kind of neurons? Odor, right? Odorant receptors. This is for the smell. Look it over here. Now, this is how your cilia will look like. For example, in this figure, let's see first figure B. What do we see? We have got the olfactory sensory neuron. Okay, look it over here. Then at the surface, okay, at the surface you have got the cilia. Now, so you know wherever your olfactory neurons are present at the surface level, you have got the cilia. So when these smell, you know, the odors comes, so they go and bind to this, where on the cilia you have got those receptors. Okay, now basically for our senses, eye ho gaya, like to see, to smell, to taste, and to get touch, right? और कौन से होते हैं senses? Okay, I'm not getting the last one. So all these are having GPCR. Okay. So the GPCR based signaling it is. So this we will be covering when we'll start the cell signaling. So here, every time please remember, whenever we talked about the senses, okay, any of it, whether to vision ho, smell ho, taste ho, touch ho, it is all GPCR based. So on this top of here you have got so many GPCR. So where your odorant, which act as the ligand, okay, they go and bind to these receptors, and finally the cell signaling takes place. Okay, that's a different story. Now here you see you have got the nucleus and the axon body. Okay, now if you remember axons, kya tha wahan pe? You have got the microtubules in the axons, which are arranged in the parallel way like this, if you remember, right? Now coming to the figure A. So A is a photoreceptor. This is for your visions, right? So here you can see that the rod cell and cone cells are there, right? Now, look at the cilia which is present in the case of the rod and cone cells. Are you able to see the difference? In this, it was like these projections of the plasma membrane, right? But here it is modified. It looks like a comb-like structures to me, right? So here, first of all, these are your membrane, these are your cell nucleus and those axons, then dendrites are there. Now over here, where the ligands, like in the case of the retina, it is the light which falls on this, right? And then it gets activated. So here you have got the connecting cilium and the modified cilium. Okay. Are you able to see the difference? Like how these photoreceptors and odorant receptors are working? This will cover when we'll start with the GPCR cell signaling. But right now, how the primary cilia functions, did you get that? Everyone? Akshaya?
Yes, ma'am.
Yeah, cool. Okay, so this is just the patch clamp recording from an olfactory cilium. So what is patch clamp? Patch clamp क्या करता है? It will just touch a small portion of the neuron and it will understand like what kind of impulses are being sent. Okay, so it is just that it is going to give you the understanding like how the calciums, Na plus, K plus, all their movement is happening and how these movements are helping in the voltage generation. Okay, eventually resulting in the nerve impulse. So I'll take patch clamp separately for you guys. Let's see this like how there is a single olfactory receptor and to its dendrite. Okay, there are several cilia present. If you can see here, line also is there. These lines like this are there. So these are nothing but the cilia. Okay, so of these, one will be one will be captured so that the recordings can be done. So how they have done that we'll watch. So these are the cilia which you can see, right? Like it helps in the sensing. Now, this is the electrode. So it just makes a, attachment over here. And then it will make a readings like what kind of impulses are being generated. And based on that, it understands like kaise particular cilia or particular neuron is functioning. Okay, so that is the work of the patch clamp. So now we know that there are motor cilia and flagella, which is responsible for the cell movement. So this is for your sperm. And then these are the like where, so we know that in the case of the respiratory tract also, so many cilia are present. So what is the function over there? What is the function over there of the cilia? So if any bacteria or any other extracellular foreign materials comes, it gets attached to the respiratory tract because the goblet cells are present, which produces mucus, right? Now that mucus is produced so that these bacteria get attached to it. And then with the help of the cilia, they remove this bacteria. Okay, so they propel them out. Otherwise, infection will happen. So now you know, like if suppose this mucus is not produced properly, in order to maintain the viscosity of the mucus, there is the function of the CFTR. Anyone who remembers the function of the CFTR? Shruti? Do you remember?
Yeah, it controls the fluidity of the mucus through chloride channel.
Correct.
As a chloride channel.
Correct. So it is cystic fibrosis transconductance receptor, right? So this is correct that it will help in the chloride channel. So what is happening in that? What happens if this is not functioning properly?
There is a buildup of mucus.
Yes, and the mucus becomes more thick. Yeah, and there is not sufficient viscosity for which the cilia it can move. So this mucus becoming thick means what? More mucus is coming and these cilia are not able to push them back, right? So bacteria which are attached to this mucus region, they start growing in that, and you get the respiratory infections then. Okay, so that results in cystic fibrosis. Okay, good. Now, so these cells are covered by numerous motile cilia. They beat in coordinated back and forth motion, either moving cell through fluid or moving fluid over the surface of the cell. Did you get it? या फिर आप पानी को पुश करो सो दैट यू गो अहेड, और या फिर आप उसको ऊपर से पुश करो सो दैट आप नीचे से इसको स्विम कर सको. ठीक है. फॉर एग्जांपल द सीलिया ऑफ सम प्रोटोजोन्स सच एज पैरामीशियम, दे आर रिस्पांसिबल बोथ फॉर सेल मोटिलिटी and also for sweeping the food organism over the cell surface into the oral cavity. So this is like आप पूरा का पूरा पानी आप swim कर जा रहे हो so that you can move and reach the food area, so that you can have your like so that the organism can have the feed. Okay, now I think yes, I have this video. So there is no sound, so you can see so many cilia are present in the paramecium, right? And look at it, like how nicely going back and forth each cilia and helping them to move in the directions. So many are there, so many. Okay, so in animals, an important function of cilia is to move fluid and mucus over the surface of epithelial cell sheets, and good example is provided by the ciliated cells lining the respiratory tract, which clear the mucus and the dust from the respiratory passages. Okay, so let's watch this is a complete video, I guess.
The air we breathe contains foreign matter such as
The voice is audible to you in this video?
Yes, ma'am.
Okay.
dust, bacteria, and pollen. Air-like cells called cilia, as well as goblet cells, can be found in the lining of the respiratory tract called the respiratory epithelium. Goblet cells secrete viscous mucus that forms a layer to trap foreign material. The wave-like action of the underlying cilia mobilizes the mucus layer, removing the foreign matter and preventing it from entering the lungs.
Okay. So now we know like how cilia is helping in removal of the mucus to which the bacteria and the foreign substance are attached. Okay. Now coming to the flagella. So flagella is also similar to structure to they are similar in structure to motile cilia, but they are longer and beat in a wavelength pattern. Okay, so cilia का हमने क्या देखा था, that it is in the back and forth, आगे पीछे, but in the case of the flagella it is like a wave-like pattern. Okay, and that's what they have shown over here. If you notice, ऐसे, right? So we make it like a wave-like pattern like this, right? In the same way, it is also moving. The cells usually have only 1 or 2 flagella, which are responsible for the locomotion of variety of protozoans and of the sperm. So we'll go in detail like how cilia and flagella also looks like. So both primary and motile cilia are anchored in a centriole called a basal body, which contains 9 triplets of microtubules. Okay, so cilia, flagella, they are from the centrioles. Okay, so basal bodies are derived from the centrioles, which has been transported to the plasma membrane. So please remember, we are talking about the locomotory functions of the microtubules. So that is cilia, and it is anchored in the centriole. So 2 of the microtubules in each triplet of the basal body are extended to form the axoneme, and basal bodies thus serve to initiate the growth of the axonemal microtubules as well as anchor cilia to the surface of the cell. Now, once you see the image, it will be clear. So this is how it looks like. So here you have got the basal body. Okay, यहाँ नीचे आपका centriole है, and as we said that it is made up of 9 doublets. Okay. Now, can you tell the difference between the centrioles present in the cilia and the centrioles which is present in the mitotic cell? Krishita? So the centrioles which we have for mitosis and the centrioles over here it is present. So any difference? Do you remember? Tanvi? Okay, anyone else? Akshaya, Shruti, would you like to give the answer?
Maybe the alignment, like how they are placed.
No. Do you remember the image like this of the centriole which I have shown? वहां पे क्या था? यहाँ पे doublet is there of 9. What was present over there? So here 9 are there. Doublets. Over there it was triplets were there. Okay. Now let's come over here. So here we have got the basal body. Now from the basal body your microtubules are extending like this. One second. Okay. So these are the microtubules which are extended like this. Okay. Now, do you remember the microvilli which we have talked about? So there it was your actin filaments, similar to that. Okay. Now, this is the cross section. So these regions are called axoneme. Okay. Now, taken the cross section, you can see that microtubules in bundles of 2, they are arranged. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. So 9, 2 are there. Sorry. Now, this is for the primary cilia. Okay, which is meant for the sensory actions. Okay. Now, coming to the motile cilia. Again, same thing, basal body is there. From there, it is extending all its microtubules. Now, what is the difference over here? That you have got plus 2 more microtubules present at the center. So, इसको हम क्या बोलते हैं? 9 plus 2 centriole pattern. Okay. Whereas in the rest, it is only 9 plus 0. यहाँ पे center में कुछ नहीं है, right? So it is 9 plus 0. This is 9 plus 2 pattern. Please remember. So for your motile cilia flagella, it is your 9 plus 2 pattern. Now, look at the diagram carefully. ये important है, याद रखना. Okay. So what do we have then? These are the central pairs. So this is your 9 plus 2 ka 2. Rest all 9 are there. Okay. Now please do remember. So there are 2 types of microtubule here, right? ये हो गया आपका A, ये हो गया आपका B. So A, we know that the microtubules are made up of 13 protofilaments, correct? In this case, you can see that B वाला जो है थोड़ा सा छोटा बनाया है, right? It means it has got around 10 to 11 protofilaments. Now, if you see, there is a protein called nexin. Protein called nexin. Nexin क्या कर रहा है? It is attaching one. This is an attachment between the AB microtubule and the another one. But how this attachment is? B का A से attachment है. Can you see? So it is a B tubule which is in attachment with the A tubule of the next. ये first का with the second का. Okay? This one. And all should be like this only. Then again A के बाद यहाँ पे B आएगा. So this is in attachment with the A again. Am I making sense? Everyone able to follow?
Yes, ma'am.
Okay. It's important, okay? Because the video is very fast. That time you won't understand कि क्या चल रहा है. Then there is something called outer and inner dynein arm. So we know what is dynein. So dynein is what it is your motor proteins, microtubule motor proteins, which walks towards the minus end of the microtubule, correct? Remember? So these are the those dynein motor proteins are there. Now when you say outer and inner, what does it mean? Of the nexin, जो बाहर की तरफ है, okay, that is your outer dynein. And then inside of the nexin, it is your inner dynein arm, and both are really very important. Okay, and now all these are in connections with the central pair of the microtubules with the help of the radial spoke. Okay, now keeping all these things in the mind, okay, try to imagine when these dynein molecules are making movement on this. So just imagine, जैसे आप किसी rod के ऊपर आप चल रहे हो, what will happen? This rod will move in this way. ऐसा कोई pipe है आपके पास, okay? Let us say that these two are those pipes only, ऐसे, right? Basically, these microtubules are arranged like this only, ना? And यहाँ पे दो pipe और है, ऐसे. Now, the dynein molecules are present like this, with two legs, inner and outer. They are walking on this. So what will happen to this? They will rotate in this direction. ये rotation समझ में आ रहा है आपका? I'm holding this one and इसके ऊपर चल रही हूँ, तो क्या होगा? Rod आपका घूमेगा, the pipe will move in this direction, ऐसे. Are you able to get this imagination? ये वाला part का?
Yes, ma'am.
Yes, ma'am.
Cool. So this is not one here. So ऐसे ही बहुत सारे हैं, क्योंकि एक से इतना बड़ा microtubule नहीं move होगा. So you have got many. Okay, everyone in sync is rotating the downward वाला microtubule. ठीक है? So when this is happening, after the moment this becomes inactive, this becomes active. तो इसके नीचे जो आपका dynein है वो active हो जाएगा, and it is gonna move the नीचे वाला microtubule. So ऐसे करते करते क्या होगा? The entire thing is rotating. Okay. Now imagine all dynein molecules are doing this thing. तो जैसे इसने किया, this gets activated, ये यहाँ पे रुक जाएगा, ये आपका inactive हो जाएगा, ये active हो जाएगा. So then this one is rotating this one. ये इसको ऐसे करते करते it is in circular motion. So you'll see that it is rotating. The entire thing is rotating, and that's how your wave-like pattern is generated. ठीक है? कोई भी चीज लो, आप उसको ऐसे ट्विस्ट की तरह गोल गोल घुमाओगे, so what happens? वो एक coil structure जैसा नहीं बन जाता है, and then if you leave it, if it is having the open end, what happens? It forms a wave-like structure, or like से propeller की तरह वो rotate होगा. ठीक है? Now keeping all this thing in the mind, let's proceed. Okay, and then that video will also make sense. Now whatever I have discussed, same thing in the written form it is given. So the axoneme of the motile cilia of flagella, it contains additional central pair of microtubules which is associated proteins that are responsible for motility. Okay, so as I was saying, so the axoneme of the motile cilia and flagella, it contains additional pair of microtubules. So this one they meant, okay, this additional one, and associated proteins which is responsible for the movement. So they are arranged in 9+2 pattern, okay, in which the central pair of microtubules is surrounded by the 9 outer in doublets, okay, in doublets, please remember. Now the two fused microtubules of each outer doublet are distinct. A, which is the A1, it is the complete microtubule which has got the 13 protofilaments. The other one is B tubule, which is incomplete and contain only 10 to 11 protofilaments and fused to your A tubule. Okay. This also we have covered. Now, the outer microtubule doublets are connected to the central pair by radial spokes and to each other links of a protein called nexin. So, आप का fuse है to your central core also, right? And also to the nexin. Now, in addition, two arms of dynein are attached to each A tubule, and it is a motor activity of these axonemal dyneins which drives the beating of cilia and flagella. So, without this dynein, there is no movement. Okay. The movement of the cilia and flagella result from the sliding of outer microtubule doublets relative to one another. यानी कि जब ये slide होगा with respect to this, then it is gonna create movement of this in this. So ये एक दूसरे को help करते हैं in the sliding, right? And this is done by the motor activity of the dynein arm, which results in the sliding. So the dynein bases bind to the A tubule while the dynein head groups bind to the B tubules of the adjacent doublet. Okay, see, यहाँ पे है आपका B, ये हो गया A, right? So जो head है वो आपका A की तरफ जाएगा, sorry, B की तरफ जाएगा. Now the movement of the dynein head groups in the minus end direction, it causes the A tubule of the one doublet to slide toward the basal end of the adjacent B tubule. Now, because the microtubules in an axoneme are connected by nexin, एक दूसरे से linked है, so एक का slide is gonna cause them to bend, forming the basis of the beating movements of the cilia and flagella. Okay, are you able to get this point written in this slide, or should I explain again? Same thing which I have mentioned earlier. So just look at this dynein. Okay, ये A हो गया, ये B हो गया, तो A के आपके पास bases है now, and the head group is present towards the B. So when this movement is happening with the help of the dynein, okay, just imagine ना ये एक पैर है, like it's they are the legs of some person, and वो they are helping कि वो इस तरह से rotate करे, तो क्या होगा, ये ऐसा rotation आएगा. So this entire microtubule will rotate since they are connected. So when this is moving, this will also move. ये move हो रहा है तो ये भी होगा. So eventually everything is moving. Okay, I guess I'll show the video. Okay, here also. Okay, here also. So you can see A है, B है, ठीक है? So dynein is present. The motor head group is towards the B. Now when this is moving, okay, the movement of the dynein head group towards the B का minus end, what is happening then? It is moving. And जिसकी वजह से क्या होगा? Everything when it is moving, so they are getting bent. So this bending of the microtubules will result in the sliding of the microtubules. Are you able to imagine कैसे हो रहा है? See, this is suppose say 1 and 2 legs. ठीक है, आपने क्या किया, 1 से पकड़ रखा है, 2 को आपने यहाँ पकड़ा, राइट? आप कैसे मूव करते हो किसी पाइप के ऊपर, अगर आप बहुत बड़े पाइप के ऊपर आप खड़े हो, सो व्हेन यू वांट टू मूव इट, यू आर गोना मेक अ स्टेप एंड देन उसको पीछे की तरफ आप रोल करोगे, देन यू विल टेक अनदर लेग एंड देन अगेन रोल इट, राइट? जस्ट इमेजिन द सेम थिंग ओवर हियर. Now why there is bend coming? क्योंकि आपने इसके आगे वाले पोर्शन को, जैसे यहाँ से आपने यहाँ टच किया है, now it is pushing over here in this direction. So अभी क्या होगा, आपका ये microtubule ऐसे रोटेट होगा. Now why this is getting bent? Because आपने अपना leg stretch किया उसको पकड़ने के लिए, so obviously onto which you are holding on, वो भी आपके साथ नीचे आएगा, and that's why there is a bend. Did you get it? Okay, maybe I can show, if you're getting it, it's really good, because then only that video is gonna make a sense, and the video is very fast. Okay, here is a video. Let me share the sound. Okay.
Motile cilia and flagella are hair-like appendages of cells that generate fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualized the molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cargo is pulled forward by the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However, if dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism generates the flagellar beat. As one side is inhibited, the other side is active. In our model, regulatory signals result in inhibited dyneins on one side. This allows dyneins on the opposite side of the flagellum to walk along the microtubules. Molecular linkers resist this sliding, which causes the flagellum to bend in one direction. By periodically switching the side of dynein inhibition, the flagellum alternates the direction of bending, resulting in the typical whip-like motion. The propagation of dynein inhibition in different regions of the flagellar wave along the length of the flagellum leads to the characteristic beating motion. The dynein inhibition may occur in an all-or-none fashion, similar to an action potential, which could explain how cilia and flagella can switch their bending direction back and forth in less than 1/100th of a second and continue beating for days.
So everyone, was that video helpful? Like, were you able to imagine like how these dynein are helping in the movement, in the sliding of the microtubule? Do you want to watch it again? Because see, if you remember all the proteins present, the nexin, about the radial spokes, and then dynein, how it is helping. So you saw that so many several of dynein molecules were present, right, throughout the flagella. So what they were saying over there was, it is not that every part of the microtubule is active. मैंने बोला ना, जैसे one part of the dynein molecule is helping in the movement. Okay, are you able to hear the voice? Internet is coming as unstable. Hello?
Yes, ma'am.
Okay. So what they're saying was that जैसे ही वो movement कर देता है, like one stretch of dynein molecule, the ATP is being utilized. Okay, the next part of your flagella is getting activated. एक bunches में उन्होंने divide किया था. So if suppose they are saying that here it is there, if these portions are active, so that time these are inactive. Then what happens? जैसे ही ऐसे वो signal pass हो गया, then this becomes inactive, the other part gets active. This active, inactive is giving them this movement of your flagella. Okay, so let's watch it again. This is really very important because this is how flagella, cilia, whatever is responsible in the movement, it is happening. And also understand that even though we are taking this much time to understand all these concepts, okay, this phenomena, but in nature, this is just happening within a second, right? Not even second, less than a second. And the sperm or the other molecule or the bacteria which has got the flagella, they just move very fast in the nature. Just watch it again, understand it, and then we'll move forward. Okay, we'll go ahead.
Motile cilia and flagella are hair-like appendages of cells that generate fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualized the molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cargo is pulled forward.
So here if you see, guys, पहले यहाँ पे अटैच है, ठीक है, अभी इसमें भी नहीं दिखा रहे, बट कैसा होता है, पहले यहाँ अटैच है, when they leave, okay, ATP hydrolysis होता है, then they attach over here. नेक्स्ट में यहाँ जाएंगे, तो ऐसे क्या होगा, ये इस तरह से मूव होगा, ऐसे पूरा मूव होगा, ओके.
By the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However, if dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism generates the flagellar beat. As one side is inhibited, the other side is active. In our model, regulatory signals result in inhibited dyneins on one side. This allows dyneins on the opposite side of the flagellum to walk along the microtubules. Molecular linkers resist this sliding, which causes the flagellum to bend in one direction. By periodically switching the side of dynein inhibition, the flagellum alternates the direction of bending, resulting in the typical whip-like motion. The propagation of dynein inhibition in different regions of the flagellar wave along the length of the flagellum leads to the characteristic beating motion. The dynein inhibition may occur in an all-or-none fashion, similar to an action potential, which could explain how cilia and flagella can switch their bending direction back and forth in less than 1/100th of a second and continue beating for days.
Okay, I hope everyone got it. Yeah, can I get the response, like if you got the idea or the concept behind the cilia and flagella, how they move? Everyone? Yes, cool. Thank you. Okay, now microtubules during the mitosis. I hope everyone has got time. Yeah, so I'll continue then. Microtubules during the mitosis, like how the microtubules are behaving in the process of the mitosis. Now, did you remember like how I was trying to explain, like when these kinetochore microtubules go and bind to the chromosome, then I had asked you a question like which part of microtubule is getting depolymerized so that the chromosomes are move or pull apart to the opposite poles, right? So that time the answer was at the plus end only the catastrophic depolymerization of microtubule is happening. Okay, so that was the answer. Now we are going to see how exactly this is happening. Okay, I try to get all the images and the video so that the concept becomes more clear. Okay, so during mitosis, dynamic instability accelerates as a result of increased rates of both microtubule catastrophe and the growth. Okay, instability matlab kya hua? Same time it is depolymerizing and polymerizing. Microtubule array present in interphase cells disassembles and the free tubulin subunits are reassembled to form the mitotic spindle, which is responsible for the separation of daughter chromosomes. Now, did you get this? So when we say that the cell is at rest, it means what? It is not dividing. Okay? So we have seen in the images like this. So आपके actin यहाँ present रहते हैं, which helps in the movement. And then you have got the ऐसे microtubules, many microtubules like this ऐसे present है, right? Now when cells are getting divided, so these microtubules will get disassembles, right? And then wherever your centrioles are present, from there your microtubules will start reappearing. ऐसे ऐसे ऐसे, ठीक है? Now, this is how it is getting arranged. Then all the tubulin molecules which are present in the cytoplasm forming the national highway, like I always used to say, give this analogy, right? So from there it gets broken down and it forms the mitotic spindles over here. So these are your mitotic spindles, and these are your chromosomes which are aligned in the same phase, right? That is same plate, that is metaphasic plate. Okay, now how these mitotic spindles are formed. So in the interphase, it is there. So you know that at the interphase, all this duplication, everything is happening, right? So your DNA is getting duplication and everything, all your proteins, enzymes, everything is getting duplicated. Now, till the time your DNA is getting duplicated, okay, by that time the centrosome is also getting duplicated. So if anyone is asking centrosome is at what phase of the cell cycle the centrosome is getting duplicated, the answer is interphase. Okay, interphase maybe you have got the S phase, G1 phase, G2 phase. Then again the answer is S phase, क्योंकि S phase में आपका सारा duplication वाला काम होता है, DNA duplication etc. Okay, I hope this is clear. And I believe everyone knows also about the cell cycle, that you have got the M phase and then interphase is there. Interphase is divided in G1, S and G2. Okay, so interphase is around 23 hours and your mitotic phase is 1 hour. This is how your cell cycle is being divided. Okay, now coming to this formation of the mitotic spindle, everyone knows about it. So once the centrosomes are getting duplicated, you can see two are coming here and here. Now here also please remember, when two are there, actually ये और ये अलग हो जाते हैं. So ठीक है, and then they become the mother. From here you will get another one and from here you will get another one. So this is like mother-daughter relation. Okay, यहाँ पे एक, now every time they are present in this direction, right? ऐसे 90 degree perpendicular to each other. When they get separated, इसके perpendicular में daughter आएगा, इसके perpendicular में daughter आएगा. So now they become the mother and you get the daughter centrioles then. Okay, is this much is clear?
Yes, ma'am.
Okay, now coming over here. So your chromatin will start condensing and it will result in the formation of the chromosome. And eventually you can see the cell membrane is also extending, right? So everything is getting duplicated. So धीरे धीरे आपके centrosomes जो है वो इस तरफ जाएंगे towards the two poles. And then you know, by that time the nuclear envelope will disappear, right? And all these chromosomes will be aligned in the metaphase and the kinetochore. Kinetochore mitotic spindle will go and bind to the chromosomes. Okay, so we have got the three types of microtubules: interpolar microtubules, aster microtubules, and kinetochore microtubules. We'll come to that, like what is the function. क्योंकि काम करने वाला कौन है? सिर्फ kinetochore, which goes and binds to the chromosomes. Then why do we need interpolar and the aster microtubules is also very, very important. Okay, so we'll come to that. So this is how the mitotic spindle is formed. Okay, now three types of microtubules. So kinetochore, astral, and the interpolar microtubules. Okay, so these microtubules now, so as already mentioned, kinetochore microtubules are going and attaching with the chromosomes, right? Now then we have got the astral microtubules and the interpolar microtubules. So you can see here the interpolar microtubules are making connections with the other interpolar microtubules. And then you have got the kinesin motor proteins over here. Then we have got the astral microtubules. So anyone who can have, who has the idea like why the kinesin motor proteins are present here? क्या काम होगा interpolar microtubules का? Any guesses? Tanvi, would you like to make a guess? Krishita?
Sliding through each other so that they can come closer and accordingly move.
Correct. So what they do is, so when this sliding is there, so this is just because of the kinesin molecule. So we know that kinesin means what? They will move towards the plus end. So इस movement में क्या होगा? They are pushing. इसको इस तरफ push कर रहे हैं, इसको इस तरफ push कर रहे हैं. And this is gonna help in the movement of kinetochore after the separation. Okay, so they will help in bringing the chromosome towards this end and this particular chromatid at this end. Okay, so that's the function of the interpolar microtubules and the kinesin. So kinesin is gonna make that push because of its movement on these microtubules. Okay, so you know kinetochore, now you know interpolar. Now what about the astral microtubules, Akshaya? Okay, let us say that this is your cell ka boundary, okay, cell membrane. So these astrotubules, what they do is that now everything needs to come closer. So this astrotubules are gonna make push from here. Okay, they have got the proteins here, and these proteins are gonna push these microtubules. And that's how astromicrotubules are making sure that they come closer and together, compact structure. Okay, don't worry, I have got another figure also, but just get an idea that interpolar microtubules and the astromicrotubules, they both are helping in the cell to bringing the chromosome to one center. Okay, ऐसा नहीं होना चाहिए कि ये यहाँ चला गया, ये यहाँ चला गया, right? Everything has to be together so that finally they can go for the division over here. Correct? What is the important thing is that we are going with the detailings over here. Okay, and this part of your microtubules region, this is important. Right now what I'm going to discuss is really very important, and same cheez carry forward hoga aapke next topic mein when we start with the cell cycle. Okay, so that's why this topic is very important, like how chromosomes are being separated. As mitosis proceeds, the duplicated chromosomes attached to kinetochore microtubules align on the metaphase place, right, equidistant from the two spindle poles. So these astral microtubules ensures that everything is arranged, or it is provided in the same distance. Now the alignment of chromosome is mediated by rapid growth of the kinetochore microtubules and capture of their plus ends by the kinetochore. So kinetochore kya cheez hai actually? So it's a complex of proteins which helps in binding of the centromere of your chromosome. Okay, so once all the chromosomes have aligned on the metaphase plate, the links between the sister chromatids, which we will learn later, right, those securine separase which is present, they help in then breaking the sister chromatids into two, then we get separately. So with the sister chromatids separating and moving to the opposite poles of the spindle. Now see, chromosome movement proceeds by two distinct mechanisms, which is known as anaphase A and anaphase B. Okay, and in which the involved different types of spindle microtubules. Now anaphase A, it consists of the movement of chromosomes towards the spindle poles along the kinetochore microtubules, which shorten as chromosome movement proceeds. So anaphase A mein kya ho raha hai? Kinetochore microtubules are getting depolymerized. Okay, now look at the figure. So in this you can see that ये आपका एक sister chromatid है, okay, to which the microtubule depolymerizing. So यहाँ पे जो आपका assembly है, so this is nothing but the kinetochore having the complex of the proteins attached, and as you see, microtubule depolymerizing kinesin, can you see this one? ये क्या कर रहा है? आपके chromatid को पकड़ के रखा है, right? उसको इस डायरेक्शन में लेके जा रहा है, but at the back what is happening, the tubulin molecules are also getting dissociated, है ना? Otherwise what will happen, this particular thing, how it will come if the microtubules are still present, ऐसे से present रहेंगे, so then it is difficult for your kinesin to bring this. Are you able to get it? So इसलिए in the nature they have done that, जैसे जैसे ये काइनेसिन आपके क्रोमोसोम को इस डायरेक्शन में लेकर आ रहा है, वैसे वैसे पीछे से आपका माइक्रोट्यूबुल जो है it will get dissociate. So there is a free path for the chromatid to be moved along the along with the kinesin movement. Did you get it now?
Yes.
Yeah. Now I'm asking a question. So when microtubules are formed, so we know that it is the plus end which goes and binds to the chromosome, correct? And we know that here it is minus end. हमें ये भी पता है kinesin is a motor protein which moves towards the plus end और dynein है जो move towards the minus end. अगर ये plus है और यहाँ minus end, then why the kinesin is moving towards the negative end?
Ma'am, there was an exception of this kinesin which would move towards minus end. Kinesin 6 or something.
Yes, absolutely correct. Very good. So there is an exception for this particular kinesin so that it can bring your sister chromatid towards the center of your cell. Okay, good. Now, same thing, you have got this kinetochore. So again, the protein complex, motor proteins are there, which is bringing your chromosome in this direction. And piche se tubulin molecules, the microtubules is getting depolymerized. Okay, now, movement of chromosomes along the spindle microtubules in the minus end direction is driven by the chromosome associated kinesins bound to the plus ends of the microtubules. So these kinesins, they act as microtubule depolymerizing enzymes as well, and that they disassemble and shorten the kinetochore microtubules as a chromosome moves to the opposite poles of the spindle. So usually what happens is that these kinesin molecules, they just need, energy driven molecule like ATP, GTP to walk on it. But ye kya hai, chromosome associated kinesin, they are actually helping in the dissociation of the tubulin molecules. Okay, so as they move, they make sure that the पीछे से आपके सारे microtubules जो हैं depolymerization के लिए prone हो जाएं. Okay, now coming to the anaphase B. So anaphase A में क्या हो रहा है? Your chromatids are getting separated and they are moved towards the opposite poles. Correct. Now coming to the anaphase B. So in this, both interpolar and the astral microtubules contribute to chromosome movement by pushing the spindle poles apart. So the overlapping interpolar microtubules slide against one another due to the action of the plus end directed kinesins, which cross-link interpolar microtubules and move them towards the plus end of the overlapping microtubule, that is away from the opposite spindle pole. Now, in addition to this, we also have the astral microtubules. Okay, so they are, the spindle poles are also pulled apart by the astral microtubules. This type of movement involves the action of cytoplasmic dynein anchor to the cell cortex. So that's what those proteins I was talking about. So at the end, ये आपका cell cortex हुआ, correct? So यहाँ पे आपके dynein molecules present है, which is helping in this type of movement. So the movement of this anchor dynein along the astral microtubules in the minus end direction pulls the spindle poles towards the periphery of the cell, coupled with shrinkage of the astral microtubules by the depolymerases. So क्या हो रहा है? जो let us say here it is present. Wait, let me make a good diagram. I'll be discussing this side only. So जैसे कि यहाँ पे आपके centrioles हैं, here you have got those mitotic spindles which is attached to the chromosome. So they will be coming in this direction, right? And then we have got those interpolar. So they will be sliding with each other with the help of the kinesin in this direction. Okay. So ये वाला यहाँ push हो रहा है, ये वाला यहाँ push हो रहा है with the help of the kinesin. Now we also have these astral microtubules, right? So these are in association with your dynein at the cell cortex. Okay, so what they are doing, they are pulling this side. ये भी इसको यहाँ ही खींच रहा है. So what will happen then? This entire thing, ये भी यहाँ push कर रहा है, ये भी इस direction में आ रहा है और ये भी इसको इसी direction में ला रहा है. So eventually this all three types of movement is helping in bringing the sister chromatid in this at this periphery और ये वाला आपका इस periphery में जाएगा. So once you have got the clear path over here, then the contractile ring is formed and the division takes place. Okay, I'll show another figure. मेरा diagram इतना अच्छा नहीं है. Okay, now over here, see in this case also. So first overlapping interpolar microtubules are there, so they will slide to each other. So this will be pushed in this direction, this will be pushed in the other direction, right? Now, in the second case, the astromicrotubules are there, which is again anchored to the dynein molecule. So they are also moving this. So what is happening? It is pulling like this. Okay. So in the final stage of mitosis, nuclear envelopes reform, chromosomes recondense, and the cytokinesis takes place. So each daughter cell at the end contains one set of centrosome, which nucleates the formation of new network of the interphase microtubules. Okay. So if you see in a normal cell, in a normal resting cell, nucleus के पास आपका centrosome present रहता है. So from where all those microtubules are again present for the movement of the kinesin and dynein, so that the cell organelles now it can be later arranged. Like हर एक organelle का अपना location रहता है. So this will be again arranged once the cell is at the interface. Okay. Yeah, so this is the I hope you know about the Drew Berry ka animation. So he is a person, like he's really very great in understanding all these molecular concepts. So he has brought all this understanding in the animation. Okay, he has also tried making all those movement of the protein by making the simulation model, etc., for the better understanding of the these proteins level actions which is happening. Okay, so just watch this video. Of course, it's there available in the YouTube also. So acha hai, and please try to understand like how exactly there is a section where the kinesin chromosome associated kinesins are bringing your sister chromatids in one direction, and that's little fast, so just give your full concentration over there. Okay, and let me share the audio here.
This visualization isolates a single chromosome at mitosis. The chromosome contains two identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle, used for orienting and guiding the chromosomes to their correct positions. The red region is the kinetochore, which works as the interface between the microtubules and the chromosome. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension is just right and the chromosome is correctly positioned and attached. The last little bit remaining red is the stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the kinetochore. These are molecular motors involved with a directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding from the kinetochore on long molecular legs are dynein, carrying the stop signal broadcasting system. When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell. This visual.
Yes. Did you get like how the proteins kinase and dynein were helping in bringing the sister chromatids to the one end of the cell?
Yes, ma'am.
Okay, cool. I hope you guys are enjoying this. Yeah, so that's all guys for today. I'll stop here. If any doubts are there, we can include that also. Anything else which you wanted to discuss? Okay, no doubts.